It's actually a pleasure to introduce our next speaker, who is actually the chair of this symposium. His experience is rich in the field of bioequivalence. His experience expands over industry, academia, and as a consultant. I've got the pleasure to work with Dr. Bloom on a different few cases where we were very pleased with his experience, his knowledge, and his input for what we've been working with. Dr. Bloom's is going to be covering a topic which is very dear and important to the generic business as a whole, and it's going to be covering the value-added medicine or super generics, like some people like to mention. The other thing is we're going to be talking about not failed but unfavorable bioresults that sometimes we obtain and how to tackle those in different ways. Dr. Bloom is going to announce some minor changes to the very interesting second day, which are going to be covering, I think, lunch times and some other appointments or arrangements that we have. So it's all yours. Thank you.
Yes, thank you very much, Dr. Tisi, for this very kind introduction. Good morning, colleagues. I do hope that you had a relaxing night and recovered completely from the effort yesterday during the day. Yeah, there are some changes, and I should start with the changes with which an offer which I presented to the organizers to save time. My offer was to skip the keynote lecture this morning, and we saved 3/4 of an hour, but they didn't accept it. Therefore, I'm standing here now to deliver this presentation. We have some slight changes. We have no coffee break after this introduction to the second day. We will continue directly with the value-added medicines section, which is very closely related to my talk, as you know. And Peter, Professor Langgut here, accepted to contribute more actively with his knowledge to the discussion and skip his presentation so that we save time. And hopefully, and it is our all responsibility, we will finish this morning by 12 o'clock, so we start with lunch at 12. And then after lunch, after a little shorter lunch, hopefully, we continue with a session which we had to postpone yesterday evening. And my particular thanks to the speakers of yesterday evening that they accepted this change and to postpone their presentation for today. So we will have a busy afternoon, but we start very relaxed, at least for you. Sit back, relax, and listen to what I prepared. This was really a challenge, and this challenge started a year ago at the last conference, where we suddenly had the idea to talk about the situation, what to do if a bioequivalence study fails. We started discussing this issue yesterday morning with the first session. We will continue it now, and everybody was pointing on me and said, well, there's also a chance to use the information from a failed study to improve the product, not the conventional way, to modify the product to have the chance for a second shot to achieve the goals of bioequivalence, but to go a very unexpected new way to learn from the study. And I was probably too tired to say no this day, and I accepted this challenge. And then when I started earlier this year to prepare it, I saw how difficult it is, and therefore I decided to tell you just a story. I think it should work. Well, that was not what my text was. Probably I put the wrong... no. My mobile was ringing at 12:30 noon. It was a sunny day, far away from my hometown, more than 10,000 kilometers away. It was in San Francisco. It was in San Francisco, Market Street, very in the center of San Francisco, sunny day, lots of people around, and I enjoyed the time there. Just a break of 30 minutes, because I was there as an attendee of the Millennium World Conference in Pharmaceutical Sciences in San Francisco, an event where 5,500 attendees were there. By the way, it was exactly the same day when Apple announced the new developments in a big event next to the convention center. So that was the situation. And I got a call from Jenny, Professor Jennifer Dressman, a good friend of mine, School of Pharmacy in Frankfurt, pharmaceutical technologist. And she said, "Oh, Henning, I need your help." I said, "Jenny, I don't know what I would like more than to do so, but let's see what I can do." And she was involved in an industry project which significantly failed. And she had no clue why. Obviously, there was no different and no reason derived from the performance of the product. Oops, that was wrong again. No. The product was simvastatin, 20 milligram immediate release formulation, easy product. Technology which the formulators use in this case has been successfully applied many cases before with other products, and nobody had an idea why the study failed. The dissolution was comparable in all media, so no obvious deviations, and 2 tablets have been administered as a single dose in fasted state. Look here, this is the result. These are the mean curves, even though my students have always to learn never trust mean curves, but they are indicative, I can tell you, and they reflect also the situation in the individual subject. Significant differences between the products, and bioequivalence could not be demonstrated. That's very obvious. You don't need statistics to come to this particular conclusion. The total exposure, AUC, was pretty comparable. It was bioequivalent for this parameter. However, significant differences in peak exposure. So there is a difference in rate of absorption. Obvious. You can see that here. And I was shocked, to say very openly, I was shocked when I heard what the Canadian CRO suggested to them. This is a reflex which you very often hear if a study failed. The suggestion is double the number of subjects, go for a replicate study design. I was shocked because how can this be a solution if you look at the data? This is not a variability problem here. If it is a variability problem, then of course it's possible to go for replicate design if it's reference scale procedure or whatever statistical approach you use. But here is a very obvious difference in performance between the product, not a variability problem. So this cannot help. You have no chance if you increase the number by 10 times and go for full replicate design. It's no chance in my eyes. Therefore, and I was happy that this wording has been announced often yesterday already, root cause analysis, that is the job to be done. This is difficult, this is time consuming, and I should say this at the very beginning. I know in industry, time is most essential for it. And normally, I can tell you, this development, this story which I tell you, took 2 years. But industry, if the study failed, they need a positive result within 3 months. would be best. I know that. Nevertheless, please excuse me if I guide you through a story which took 2 years, just to indicate what chances may be also in failed studies. That is my intention. I don't ignore reality. I know the situation in industry. But here, I asked for certain investigations, and the situation, that was really something which shocked me again. I went to the company, we had a discussion, and they had some irritation. That was obvious for me. I could understand. If a result was so significantly deviating from expectations, you are irritated. However, they had also suspicions. There was certain fight. I was not aware of that, but I recognized it very rapidly. There was a fight between the formulators in the company, the pharmaceutical technology department, and the clinical department. And the pharmaceutical technologists who said, we did our best and we have lots of experience of positive results using this particular technology, so we have no idea why this happened. So they are guilty. They should have done something as an act of sabotage in-house. I was shocked. I said, this is impossible. But nevertheless, Jennifer has been included, and some others as well, experts in the field where I have only limited expertise. I'm coming more from the clinical side. So people like Peter and others, they supported that, and we looked for some hints why this happened. This misperformance needs an explanation. And the suspicion was a conspirational in-house act of sabotage. And their idea was, look, there were 2 tablets given at the same time. Let's go back to the previous slide. Here, if you look at that, here, the first tablet has been administered, and the second. So their idea was, our clinical, they didn't call them colleagues, the clinical guys, they administered the tablets not at the same time, 1 at 0 and 1 after 4 hours. And then you receive such a result. So that was their silly idea, or my goodness, I should learn the technology first, though it's not too difficult. Hope that it's better now. So that was their idea. But I suggested, let's go for detailed analysis, and I said already yesterday, my intention is always to give the individual results the chance to tell us their story. Why do they behave differently? What happened in the GI tract? What happened with the formulation of the product performance in vivo? So that was the intention, and I can make it very short at this stage. The outcome was that consultants' conclusion that were more than myself present and involved. There is no evidence, there's an evidence for formulation failure, but this evidence, the reason for that could not be discovered, and there's no promising suggestion for product modification and reformulation. We have no idea what to do to improve it. Derived from the data on the table. And therefore, the R&D head finally decided, he was also irritated because of the situation in the company. He said, well, I would like to know and exclude misconduct of the study, and therefore they asked for a repetition of the study. And they asked us, SRD, that stands for Socratic R&D, that was the company I had started a few years ago at CRO in Germany. But we had a collaboration with a site, and we had a site in Brazil, and therefore they asked us to repeat the study with the same concept and design in our unit in Brazil, and the parent drug and active metabolite have been determined, and they gave us 2 alternative formulations to be tested, and you see here this is the outcome. More or less the same. So we confirmed the results of the Canadian study, principally confirmed. There's a systematic deviation between test and reference. So what we could exclude at this stage was fraud or misconduct or sabotage in-house. So this could be excluded. But this is not the solution. So why the hell do they perform so differently? So this was still an open question, and therefore root cause analysis 2.0. So we had to continue. And I was so happy that Peter, yesterday, in his second talk, which was brilliant in my eyes, because I think brilliant in particular, because he mentioned the point which I did not expect, but which is exactly what I had in mind in those days, the importance of disintegration. And some of those who had the chance or the stress to discuss in a consultancy meeting with me, they learned that I think disintegration is sometimes, or in many cases, more important than dissolution and gives you a better insight. So this is a case of that. So first of all, the BCS classification of Symposium. Again, it's my fault, probably. I don't know why. I should probably stay here. So, it's a class 3 drug, but I would like to be closer to you and have a view here. It's a BCS class 2 drug. It's very poorly soluble in all relevant media, and on the other hand, it's complete and completely and rapidly absorbed from the GI tract, from the intestine. In the stomach, no absorption occurs. The 0.5% in some cases is negligible. Absorption only occurs after gastric emptying. You should have that in mind. And you see the very rapid absorption with a maximum plasma concentration observed after 1.5 hours. The dissolution testing in all media containing a certain amount of SDS to improve solubility was very rapid and no difference could be identified. No relevant deviations in fact. And I suggested here to test disintegration. And the result was not very impressive for them. From my point of view, they were significant. We found a deviation in the normal testing apparatus, and we did not have these fancy equipment which Peter mentioned yesterday, and we did not have an idea that the media and, for example, all the components he is adding to it have an impact. So it was the classical pharmacopoeial test, and we found a difference in 6 minutes in case of the test product and 17 minutes for the reference. And the majority of scientists would tell you, negligible. Mean gastric emptying time, 30 minutes, which is wrong. They say, everything occurs in the stomach. They misunderstood that, first of all, and that's really important, and this was also shown yesterday, if you extrapolate from in vitro to in vivo, there's a significant retardation. So 10 minutes in the laboratory means sometimes 30 or 60 minutes disintegration time in vivo, in particular because of the other media which are relevant there. So this was the result. I was not really pleased, but I think, now we find a difference between the product. And the consequences are, this is important, after disintegration you don't have a solution, you have a suspension. And therefore we need to have a look how are tablets non-disintegrated tablets are emptied from the stomach, and how are suspensions emptied? And that is a very significant difference. I'll show you in a minute. So that was the first result where I then said, well, that is an interesting finding. Now, how to understand biopharmaceutics? This is a more general approach now. What do we learn from that? First of all, disintegration and dissolution in the GI tract is much slower than in vitro if you don't use these fancy equipments and media. In the stomach, and that is the second thing, in the stomach you have sufficient amount of water. You have residual volume of around about 30 to 50 milliliters, and then administration together with 240 mils. Then you have round about 300 milliliters of water. That is sufficient for disintegration. However, this is a dynamic process with a gastric emptying of pure water with a half-life of 20 minutes. So in other words, after 20 minutes you have just 150, so it's rapidly decreasing. However, you have sufficiently amount of water at the time of administration, but low agitation. On the other hand, in the intestine, there's a significantly more retarded performance due to the lack of water. In the entire small intestine, actually, in your intestine, my intestine, you may expect between 60 and 100 mils. So much less water. In this very long 5 meters small intestine, you have just less than 100 mils. So if something happens there, you have not sufficient water. This is a secretion and reabsorption process, because we know 12 liters are going through the intestine per day, but it is not, 12 liters are not available in your intestine right now. It's just a little bit only. Now, and this is very important. I show you the outcome. We did for almost 10 years imaging studies in the GI tract. We were interested in a research program together with Professor Weitschis from Greifswald in Germany. We were interested in the interaction between GI physiology and performance of dosage forms, in particular modified release dosage forms. But we also learned from all these studies, and I looked at 150 of those studies, and I looked what was the time of gastric emptying of those tablets which did not disintegrate before. Modified release formulations only don't disintegrate if they are not polydispersed forms. And you see here, and that's important, 25% of tablets which do not disintegrate in the stomach are emptied within 10 minutes. So after 10 minutes, 1/4 of the administered tablets have the chance to be emptied from the stomach in fasted state. Another 25 or even more than 30% in the next 10 minutes, and another almost 25% after the next 10 minutes. So after 30 minutes, more than almost 80% of the tablets which did not disintegrate are empty. And if you now remember from the laboratory, 17 minutes disintegration time. So these tablets have a much higher chance, because it's delayed in the in vivo, to be emptied without disintegration than the test product, which disintegrates significantly earlier and has a better chance to disintegrate in the stomach and form a suspension. That is the consequence. Just to, I can tell you a lot, and I can test my credibility in the audience. However, I can also show you something, and I would like to show you a short video which shows the by far fastest gastric emptying of a tablet which we ever observed. This is the video sequence which I show here, the tablet in the mouth. This is the esophagus. Here it enters the stomach, and here's gastric emptying. So you see in this sequence the tablet going down, it is marked magnetically, entering the stomach, and here leaving the stomach. And you will see this tablet, which I show you in the video, will be emptied in 5 seconds. 5 seconds after starting swallowing, the tablet arrived in the duodenum. 5 seconds. Now, let's have a look. You see, now this is entering in the stomach, and this is gastric empty. Here is the duodenum after 5 seconds. So it can sometimes occur very rapidly, and such a tablet has no chance, even the fastest disintegrating tablet, to disintegrate in the stomach. It will disintegrate in the intestine. And that is sometimes the reason why we have outliers or some unexpected results. Because something happens which we do not expect. We expect 30 minutes, or I would say 20 minutes, mean gastric residence time. But it's mean. Never trust mean curves. And sometimes it happens much more rapidly and sometimes later. Here you see all are emptied after 2 hours. So that is what we observe. And now, how are suspensions emptied? I show you here, that is a very nice example of paracetamol solution. Perfect. Individual curves. The mean curve is representative for the individual ones. Very nice. And that is a suspension, because diclofenac effervescent tablets is a solution if it's ingested. However, in the stomach, acidic media, the diclofenac is precipitated, we have a suspension. And here you see what happened. These are all gastric emptyings. Look here, first gastric emptying of this suspension. Second gastric emptying here. Third gastric emptying here. And also other cases. You see the suspension is not emptied at once, because the stomach is not a dissolution apparatus with a pedal, mixing everything fine. No, it is sedimentation, and suddenly a portion is empty, and then you see rapid absorption. So that is the difference which occurs in case of suspension. So the lessons learned is the deviation in performance might be due to the difference in tablet disintegration. The first time we saw, maybe this is the reason. And tablet disintegration in the stomach forms a suspension, which is emptied portion-wise, absorption from the intestine very rapidly, and therefore you see multiple peaks, and the resultant in the mean curve shows you 2 peaks. And the second peak is related to the intake of the next meal after 4 hours. Therefore, you see the increase in particular after 4 hours, because the rest of the suspension, by taking the next meal, is pushed out of the stomach. That's the reason why most of the second peaks are around the time 1.5 hours after the meal intake. So the portion-wise emptying, that's and the tablet. If the tablet disintegrates in the intestine, also the suspension is formed. However, in a very small area, and in this particular area, now water is secreted, dissolution occurs, and immediately the dissolved active is absorbed, and therefore you have a more harmonic profile as seen for the innovator product. So what are the consequences? Of course, the consequences is, that's the obvious reaction, reformulation. That's essential, as long as bioequivalence and generic development is the goal. However, I started to convince the company to go an alternative way. And our reflection was to discuss what is the clinical meaning of this totally different profile. What do we expect clinically if we compare the innovator product and the totally different, more retarded profile of the reference product. A very strange suggestion. However, that was in a time when out of the blue, 2001, the lipobuy case came. Cerevastatin. You may, the older colleagues will remember that. That was in the USA, US FDA, this brought the approval of lipobuy. And lipobuy has been introduced as the statin with the highest bioavailability of all components, very good efficacy, low interaction potential in the clinical setting before approval, except gemfibrocil, which was declared a contraindication. Nevertheless, due to rhabdomyolysis cases, it was withdrawn from the market. In particular in the US, I have to say, because very often the contraindication was not considered, and the majority of the rhabdomyolysis cases, and there were about 50 people dying from that, in the majority of cases it was, I think, 80% was due to not considering and not respecting contraindication. But nevertheless, that was the situation. Why do I mention that? Because this was or initiated basic research of statins, and what did we learn from that? First of all, there are two sides of efficacy. One for the desired effects and one for the undesired. So the site of action for the desired effect is in the hepatocytes. That's the place where statins develop their mechanism of action, and the extent of exposure in the hepatocytes is most essential for the desired clinical effects in lowering cholesterol level. Determinant for safety and adverse events is, on the other side, the peak concentration in the central circulation. In the muscle tissues, this is the site of action for the undesired effect. So two distinct sites of action which are in different parts of the body. And the conclusion and the development consequences, first of all, the therapy of statins is undoubtedly life-saving, but not without any problem. And there's room for improvement, and this is desirable to optimize the risk-benefit relationship. And the idea was that we target the product to the site of action in the hepatocytes and reduce the concentrations in the bloodstream. And there was an essential finding of those days, and that finding was that statins are absorbed or taken up into the hepatocytes by a selective transporter-mediated mechanism. That's the OTP1B1 transporter pumping the active into the hepatocytes. And this access to the site of action should be separable, because proteins are available only in a limited number of these transporters. And therefore, if there is a high number of molecules, they all are separated, and the rest is will be transported into the central circulation. That was our starting point. And the consequences in our theoretical assumption at this stage for product improvement was that we wanted to reduce the statin concentration in the central circulation, the muscle tissues. We wanted to do this by retardation of drug absorption, for example, by a sustained release formulation, in order not to separate the transporters, and the expected effect was a reduced separation of the transporter, more transport into the hepatocyte, less into the central circulation. That was the theoretical concept. And fortunately, we were very happy about that. The company said, well, that's not a lousy idea. Seems reasonable. And they said, okay, let's go that way. And so, just to explain it to you with this animation, this is are the transporters. They are pumping the balls. The yellow balls are the stated molecules into the hepatocyte, and here is the mechanism of action, the HMT reductase, which is blocked by the statins. And if you reduce, now there are 7 balls, if you and you see the result, that's the exposure which you measure. If you reduce the number of balls coming to the transporters per time, let's say to 5, for example, then you see a reduction of the plasma profile. So exposure goes down. That was our assumption. And this was, on the other hand, the development concept. Now, so the expectations were improvement of efficacy, safety balance, sufficient exposure at the hepatocyte that's needed for efficacy, reduction in systemic bioavailability intended for better safety. And now you see the results of proof of concept study. This is the modified release formulation, 60 milligram, because we were not so sure whether 20 or 40 would be sufficient. But we saw this significant reduction between IR and modified release. So the bioavailability of the modified release formulation was just 50 to 20%. But that is not sufficient. It could be a lousy formulation where the drug is not released. So is that due to our expectation based on the mechanism of action? Is the majority of the active molecules pumped into the hepatocyte? So we needed a clinical proof. And therefore we continued with the clinical studies in patients with LDL cholesterol reduction. Such studies normally start with 4 weeks run-in phase, where, by the way, the majority of people drop out because they are not really need to be treated, but just to diet, it's sufficient to reduce their cholesterol levels. But anyway, so then the 180 patients were enrolled, and you continue with active treatment over 5 weeks. That's the normal procedure. And you see here we already reduced the dose of the extended release to 40 milligrams. So we had 40 milligrams of this blue product and 60 milligrams of the immediate release reference product. And you see there was no difference in efficacy. So there was an improvement in efficacy shown by these data, and the consequences, there's for sure same or better efficacy despite lower systemic exposure, and this was the rationale for this development and the confirmation of our concept. That is a little bit end of story. I have to say, to be very open, this development was stopped at this stage. We planned already the next step. However, altogether it took too long. We were already now 1.5 years before patent expiration. And therefore the company said we will not be able to come on the market with the improved product at the time of patent expiration. That was our intention, that was our dream. And we wanted to come with an alternative, with a better alternative at the same time, and therefore we decided to stop that, unfortunately. However, I think it is an example where we can learn something from BE failures to improvement. What do we learn? The bioequivalence study failed, and the consequences are normally root cause analysis. This is essential, and then to find out why it happened. And if you find a formulation failure, then you can go for an improvement of the product, but not just as a reflex with each study with more products, because this is the only way to go if there's a clear indication that variability is a reason for this deviation. Or use the clinical implication of the deviations which could be verified. It's a chance for improvement. As a final remark, I would say, I cannot go more in details, but that is the message for today's discussion. And the program for something like that, and that's bad news probably for those who have the dream, oh, that's very easy, and that's nice, and that's sexy in a way. Yes, it is, no doubt. However, if you go for an added value medicines, you have a certain clinical program which has to be considered, because if you claim advantages, you need to demonstrate, to prove these advantages. There is evidence needed for that, and the bioequivalence assessment can not suffice, because if you show equivalence, you cannot claim advantages. That is a contradictio per se, what I learned in my Latin schools long ago. So it is impossible. So you need to demonstrate clinically the advantages. But I think not a full clinical program, which is normally what authorities well, we have a nice guideline, you have to follow this and that and that and that, full clinical program. I think no. And I would like to fight with them with a very good clinical arguments. Why an intelligent approach with certain bridging to existing data from the existing product may help, and you should end up with a reduced clinical program. However, you have to have good arguments to convince our colleagues on the regulators' side. They have to do their job. They have to protect patients' situation. However, as scientists, they are, and they should be open-minded to arguments, and we should be intelligent enough to present a concept which is convincing for them. So that is the way to go, but we would need a full day to discuss that more in details, what clinical studies should be necessary, and it is always a case-by-case situation. So I would like to end my presentation right now. You could probably get an impression why I had some stressful days in preparing that to fulfill the expectations of my colleagues, and they pointed with the fingers on me. Thank you very much that you accepted my invitation to follow me on the way through this story, starting in San Francisco and ending now here at the Dead Sea. Thank you very much.
What an exciting presentation to start our second day of the symposium. Thank you, Professor Blum. As usual, I'm not surprised actually. You made us like very enthusiastic for the second session of VAM. That's a very good start actually to think of science, even to justify any results that we get. Because we're running late, I might take one question for Professor Blum and then move to the second session right away. Any question? You decide. One of you.
My question just to Dr. Blum is just a question about that. And while going through the whole history and the root of synthesis, we were said, all of us, and it was concluded that the formula is there's no problem with the formula. But I believe that the disintegration is part of the formula, where the disintegrate either working or not working, or the amount. So how it have been retarded or whatever, the slow disintegration without changing the formula. Just this is really what was thinking all through the presentation. Thank you.
Yes, it's always, as I already said, it's a case-by-case decision. There are, what we learned over the years, there are certain compounds which are most critical. And the most critical from the biopharmaceutical properties, the most critical in my eyes are those components which are easily soluble in acidic media but low solubility in neutral media. Because if such products are emptied, undissolved into the intestine, then you have really a problem, because the environment is very bad for them to be dissolved. So in those cases, if disintegration does not occur completely and the tablets are not formed or transferred into a solution completely during gastric residence, then you have a problem. So that's the most essential. Those who form a suspension is second worst, because suspensions are emptied differently to not disintegrate to tablets. And that was, in my eyes, the key to understand what happened there. Because if all of the tablets, let's say 50% in one case and 10% in the other case, are emptied, undisintegrated, then you have a difference in performance. In one case, you have the suspension, which is emptied portion-wise, it is something like a modified release formulation, a retarded formulation, because every portion which is emptied from the stomach is then dissolved and then absorbed. So you absorb it in small portions and you find in the mean curves some retardation. So this is the second. And so again, what I like, and Peter and myself, we said yesterday, we have to we have something to be discussed after the conference to bring all the examples together. And that's the reason why I was so excited after his presentation, because he brought other aspects which we did not consider in those days into the discussion. That means excipients. I was always saying, well, excipients are a bit overestimated in their importance. Since yesterday, I know, no, that's not the case. And also Nilofer's presentation, we will take up that point in Washington in December. I think this is the first time I see a rationale for this changes in immediate release formulation, because I said food effect is always either substance related, first class metabolism, that's substance related, or formulation related. And formulation related normally only in case of modified release form. But you showed us example, Peter, there are examples due to excipients of immediate release formulations in the laboratory which behave differently in vivo. And they are, let me say it this way, there are some kind of modified release formulations. Is there gelling effect or something like that? So in those cases, there's a good rationale to ask for FET studies as well.
Thank you.
Yeah.
Moving away from one-size-fits-all to a much more tailored and patient-centric approach. Value-added medicine is the approach to value-added medicines is emblematic of the innovative concepts underlying the new age healthcare. Value-added medicines offer unique opportunity to deliver innovative treatments to patients at a fair price for patients and healthcare systems, while at the same time recognizing R&D investments by the industry. In addition to the competition they bring, value-added medicines will also improve the cost effectiveness and efficiency of the healthcare system, either in terms of improved clinical outcomes or improved patient cares. Today, you will hear from 3 distinguished speakers from academia, consulting, and regulatory health authorities about the scientific and regulatory aspects of value-added medicines. We hope that you'll find this session to be insightful and thought-provoking. Due to time limitations, there will be no Q&A sessions at the end of each lecture, but please do feel free to engage the speakers offline during the sessions, and I would ask the speakers to please stick to 25 to 30 minutes for their presentations. So for the first lecture, it is my pleasure to introduce Dr. Attila Inkal from Turkey to speak who will be speaking to the topic of patient's desire, physician's expectation, and industry's perspective. Dr. Inkal.
Good morning. After Henning's talk, I will go on value-added medicines. But before starting my presentation, I would like to thank the organizers, Dr. VAM and also Dr. Mohamed Khalil, for their kind invitation to the 4th Symposium. What is the presentation outline? Let me give you some idea first. I will talk about the introductory part, value-added medicines, driving forces, definitions and nomenclatures. What are the value of value-added medicines? Drug repurposing, repositioning, reformulation, combination, and patient's desire, physician's expectation, industry's perspective, and going on actions by the hand of different parties on value-added medicines. What are the driving forces of value-added medicines? Let us give some short information about them. Pharmaceutical environment, you know better than I as the industrial people. Every day we have a new challenge on our table related with the pharmaceutical issues. Healthcare system inefficiencies related to medicines. Another one is pharmaceutical business model is time limited, particularly for new molecular entities. What are the current obstacles for adoption of value-added medicines? Let us give some information. You know, during the years, the new molecular entities, starting from the mid of the 90s, decreased until today, we can say. During the last few years, it started to increase again, but you can see here the increase, not conventional type new molecular entities. biological applications, license applications increasing. Now, after this changing within the last 2 decades, the most important thing, the medicines price, because of the new molecules, always increasing. But on the other hand, we have in our hand very powerful still. We have very powerful molecules can be used to treat for the treatment. Here, the value-added medicines came on the table to be discussed. Value-added molecules are based on known molecules that address healthcare needs and deliver relevant improvements for patients, healthcare professionals, and or payers. It is said that value-added medicines can improve patients' everyday life. And as you know, the active pharmaceutical ingredient in these molecules are not new molecules mostly. What are the driving force, again, for these value-added medicines? Let's continue. The benefit of value-added medicines applies to a large variety of therapeutic areas. For example, asthma and chronic obstructive pulmonary disease are, as an example, 2 conditions where value-added medicines can deliver relevant improvements. In Europe, only in Europe, 68 million people suffer from these 2 common respiratory diseases. Asthma and COPD are respectively responsible for 250,000 and 1.1 million annual hospital admissions, and their estimated annual economic burden in terms of direct and indirect cost amount to 82 billion in total. For that reason, the parties related with the pharmaceutical development and the utilization of medicines decided to review what we have in our hand. We can describe under repurposing this action. Value-added medicines, firstly, can be categorized for the special purposes. I will give here only 3 important ones, but in the nomenclature part, you will see more. Repositioning, launch into a new indication. Existing molecule can have a repositioning into a new indication. Reformulation, change in formulation, strength, route of administration. Combination or incremental innovation, 2 or more products offering into one product. Medicine device, or medicine service. As I mentioned in the previous slide, I gave only 3 of them, very important for industry and academia. But on the other hand, if we go to the nomenclature, we can see different descriptions under super generics, premium generics, specialty generics, re-innovated generics, new therapeutic entities, enhanced therapeutics, including into the combinations, improved therapeutic entities, incremental innovations, including into the combinations, re-innovation, hybrid products, biosuperior products, third sector products, drug repurposing, drug reformulation, drug repositioning, drug reprofiling, drug reusing, or drug rediscovery. Let me talk a little bit on the main three different approach. Drug repurposing includes all the development strategies based on the same chemical structure of the therapeutically active ingredient as in the original product. Drug reformulation is, by the simple definition of the term, making a particular change in the formulation of the original drug. This can be achieved by exploiting advances in the formulation technology to change the release of the active substance, pharmaceutical forms, and or route of administration, but it can also concern some excipients with no impact on the pharmacokinetic parameters. No change should be included in the structure of the active pharmaceutical ingredient except when it is a chiral switch. Cases where the development of a new product does not include a change in the original formulation. Drug repositioning, process of finding a new indication for a drug or compound. New indication is distinct from the already approved or intended indication of the original product, where distinct implies an anatomical or an therapeutically distinct indication, referring to the 10th version of the International Classification of Diseases. The situation where the new indication involves a different pharmacological target is the only exception where a new use in a similar indication will be covered by the actual definition. Let's continue incremental innovation, closely related molecules with the different attributes that may offer significant value in the treating particular disease, variants, or patient segments. And if we go different type of incremental innovations, we can see new dosage form, which affect the dosage route, dosage form, and the dose amount new formulation. The second one, how the chemical in the drug are combined to produce the drug. The third one, new combination, creation of combination drugs from existing molecules. Fourth one, new indication, using an existing drug to treat a different condition. The fifth one, new active ingredients. Drugs that contain the same active moiety but include a different enantiomer, racemate, salt, ester, complex, chelate, or clutarate, also referred as the adaptive innovation or marginal innovation. We should not forget that value-added medicines are the medicines where innovation is applied to an off-patent molecule, delivering enhanced value to patient and the relevant healthcare stakeholders. What kind of improvement value-added medicines may bring to the society and the parties, of course? The value added to an old medicine could be any of the following, and the related improvements contribute to the sustainability of healthcare system through better adherence, improved levels of safety, efficacy, and tolerability, better efficiency of healthcare professional resources, treatment for new indications or patient subpopulations, improve treatment administration, improve cost effectiveness, amongst others. Repositioning, if you take a look at the repositioned medicines are often developed and launched into new indications where they offer an alternative to existing treatments and address remaining unmet needs. We can give an example to repositioning. Impunir is a prolonged release tablets, different strengths, which is guanfacine hydrochloride by Shire Pharmaceuticals, UK. A medicine that was redeveloped to provide sustained release, repositioned to demonstrate efficacy and safety in attention deficit hyperactivity disorder patients. This repositioning provided an important alternative treatment option in the therapy area where different patients responded differently to different treatment types and met a significant unmet patient need for different mechanisms of action. Reformulations, a range of important benefits via this way can be provided to patients, to healthcare community, to society, through allowing administration higher or lower doses as by passing first-pass metabolism, delivering molecule medicines directly to the site of action by reducing blood level spikes through better efficacy, safety, and tolerability can be delivered. We can give another example to reformulation. Abraxane. Abraxane, 5 milligram per ml, powder for suspension for infusion. Paclitaxel, formulated as albumin-bound nanoparticles by Celgene, Europe, UK. This is a medicine that redeveloped target delivery of higher doses, resulting in improvements in efficacy in metastatic breast cancer, 30% versus 16% of women responded to treatment versus conventional paclitaxel, and in pancreatic cancer, an improvement of 1.8 months in overall survival as demonstrated versus gemcitabine. Provided a reduction in treatment time from 3 hours to 30 minutes. Eliminated the requirement for pre-medication as steroids and antihistamines. Let us come to the combinations. Combination products can be classified in 3 different groups. As I mentioned before, medicine-medicine, medicine device, medicine service. What are the benefits of combination products? Bring a synergistic effect, combining the benefit of medicines, device, and or services to improve medicine delivery offer, improve patient convenience, and depending on efficacy, ease administration of the medicine. What are the benefits of combination products? Let's continue. Reduced self-administration errors, and depending on these, patient and healthcare professional-centric significant improvements. Clinical efficacy improvement over the comparators. The combined value-added medicines may demonstrate significant clinical efficacy improvement over the comparators, which can be challenging for manufacturers to demonstrate at launch. example of this combination issue, dual recipromax medicine device combination and Targin, a medicine combination on the market. What Professor Tubi took onto the table, value-added medicine means rethink, reinvent, and optimize medicines, improving patient health and access. This is really very important, particularly for the all parties, particularly for industry and also academia. As a pharmaceutical technology professor, I can frankly say that more or less since 30-35 years, I was warning to the industry, hey, or my colleagues in the universities, we can use what we have in our hand as medicines, changing the delivery ways, reformulating and reducing the amount of the substance active in the drug products, improving the utilization of these substances by body, using different delivery system in our development studies. But we lost time, frankly. Now we are so close to end, and now today, value-added medicine, via value-added medicines, we will have this topic again, particularly to research and development of the industry departments. The value delivered by value-added medicines based on three repurposing models: value delivered for patients, value delivered for society, healthcare provision and organization, such as contributing to sustainability of healthcare system through economic advantages. All the provided values are based on patients' desire, physicians' expectation, and in this industry's perspective. Where is patient's perspective or desire? What does a patient really need about medicines? We should try to find the answer when, if we would like to introduce a new molecule formulation using existing molecules. What are the interpretation of patients for risk minimization? Comments on available tools for risk minimization. How patient contributes to the pharmacovigilance practice. How health literacy may affect on the patient's needs and desire. These are important issues from the side of patients and also, of course, the other parties. What is the society's perspective and expectation? Reformulations can result in optimized administration and improved patient experience, as a long-acting tablet allows patients to take the medicine once instead of twice daily. A liquid form allows patients less able to swallow tablets to take a medication. Reformulation resulting in improved patient experience resonate with physicians and patients. They tend to have limited impact on payers without a clear improvement of clinical outcome, of course. Industry's position: drug developers can offer improved level of safety, efficacy, and tolerability through drug reformulation. I will give here an example. Change presentation form by changing the presentation form of which is used in the treatment of opioid dependence, from present sublingual tablet dosage form to buccal film. One of them is placed on the tongue, the other one between the gums and the inner cheek. The buccal film provides an around-the-clock treatment, which cannot be adequately controlled with the tablet dosage form as alternative. These two forms are absorbed by the mucus, while the film dosage form offers improved bioavailability and active pharmaceutical ingredients absorption rate is between 45 to 65%. The sublingual tablets absorption rate is between 19 to 29%. The buccal film also comes in several doses, so physicians can best match the individual patient's tolerability. The reformulation and the real-world evidence data. Let's take a look at it. Improved convenience will have a positive impact on patient compliance to medication and thereby improve patient outcomes. This is one kind of patient desire. But it should not be forgotten that these aspects are difficult to prove at launch without the support of real-world evidence data, as Henning mentioned in his previous talk. This is the industry's position and need. It must be considered that industries, as already have seen by some examples, reformulation studies certainly will be helpful to illustrate the benefits and recognition of long-action reformulations. This is also industry and the patient benefits. What is the importance of repositioning? Industry must pay attention that value-added medicines could address new indications, patient subpopulations. These options effectively increases the treatment options offered to patients. This strategy is essentially drug repositioning, whereby a known medicine is giving a new therapeutic.
Excuse me, Dr. Hinkel. Could you wrap it up in 5 minutes, please?
Yes. One of the good examples to repositioning is an extensive anti-malarial drug. This drug was potentially be repositioned by researchers working in this field to treat Parkinson's disease, which currently has no cure. Combining medicines with new services, devices, and the others. This related with all parties, more or less. Drug developers can also add value to existing medicines by improving drug administration, which can be achieved by combining medicines with the new services, devices, or even other drugs. Added services have been particularly rampant given the current health environment's active shift from product orientation to service orientation. For added devices, an example is repackaging an injectable solution into a pre-filled syringe. And here we can see combining medicine with other drugs. The industry already started to go in that way. This is the additional benefit of patient, of course. And one of the big pharma company created drugs for neglected disease initiative, which led to the development of a medicine that combines two anti-malarial drugs, artesunate and amodiaquine, by enabling patients to take 2 tablets instead of 8 tablets a day. This new combined medicine was more convenient for patients and thus more cost-effective to the payers. for the payers. Out of this, I would like to give a short information what is going on today by the hand of different parties on value-added medicines. Some important actions and recommendations going on to settle down or install the full value of value-added medicines by European Union initiatives, key collaborations between the regulators and health technology assessment bodies, and other European projects involving various stakeholders. If you follow closely these developments, we can collect some new ideas. And the conclusion, essentially, added services, new devices, drug combinations can help patients better manage on their own, improving adherence and the health outcomes, and creating efficiency for the work of physicians and practitioners. Pharmaceutical and life science companies are confronted with increasing competition while working with limited resources, driving the need to improve their internal capability to innovate. Developing new innovative products, however, requires investment, commitment, time, and product potential. How can companies simultaneously work within budget and deliver innovation and value? This is the one of the pushing power value-added medicines. This dilemma is fueling the need to identify and eliminate all possible pockets of inefficiency. In product development and healthcare delivery processes, there are several of these pockets. In surveying their product portfolio, researchers may discover opportunities for innovation sitting idle and untapped in existing therapies. And by rethinking, reinventing all products that have gone off patent, companies can develop value-added medicines and optimize the benefit they offer. Thank you very much for your attention. If you have any questions.
Thank you for the informative presentation, lecture, Dr. Nico. I believe we may have time for one quick question before we move on to the next speaker.
So if we are doing like a repositioning for a drug to have a new indication, should we like reestablish the whole clinical profile by doing phase 1, 2, 3 clinical trials? Or maybe one randomized clinical trial could be sufficient to prove the new indication?
Well, it is not necessary to run for all anti-parasitic ingredients in a new combination, for example. But we should, as I mentioned in this report, we should review very well, and if we can use support of the literature, existing non-clinical and clinical literature, we can use it for our combination. But of course, what we are claiming, having a completely powerful product than the similar ones, we should run the clinical studies and show this advantage or benefit of the product. And during this period, each phase of study or the other is necessary.
Thank you very much, Dr. Hanko. It's my pleasure to introduce our next speaker, Dr. Louis Kikawi, ex-US FDA reviewer and Saudi FDA consultant, who will be speaking to US FDA's first experience on 505(b)(2) applications and requirements.
Hello. Thank you, Dr. Suleiman, for the introduction. It is my pleasure to be up here on the podium again to discuss a relatively different topic from what we discussed yesterday. And because of the interest of time and the changes in the program and the request of the organizers, I shortened my presentation, and basically it will be a refresher on what was discussed last year by actually Dr. Suleiman on the same topic. And basically he had presented the regulatory and scientific concept for development of value-added medicine under the 505(b)(2) route for the US experience. So basically what I did in the presentation was I gave a little bit of summary from the guidances, and then I am providing some example of some notable 505(b)(2) approvals that have come through this pathway. So we'll talk about some requirements and some applications. My references are, of course, the two guidances that are in effect in the US FDA. The first guidance is actually a really good guidance because it guides you on whether you should go through the ANDA route or the 505(b)(2) guidance. And then the requirements you need for the 505(b)(2) is in the second guidance. And then the next two references are pretty much publicly available literature documents that discuss the same topic, and I will be giving some examples from the 7 noteworthy 505(b)(2) submissions. So in the guidance, one of the US FDA guidance, there's a quote that says, "It is wasteful and unnecessary to carry out studies to demonstrate what is already known about a drug." So this was the basis for when the Hatch-Waxman addition to the Food and Drug Act. If we already have the information, why recreate it or why make somebody else generate it again? And this was the sentiment of Dr. Paul Hartman, who was the acting director of the Center for Drugs and Biologics in 1987. This sentiment is still true today, and we see both the EMA and US FDA are following this sentiment using value-added medicine or application through the 505(b) route. So what is a 505(b) application? A 505(b) application is one that contains full reports of investigation of safety and efficacy, but where at least some of the information required comes from already approved information, or where the applicant of that product does not have a right to reference that product, but they can use that information as it's available. So what does it mean, previously approved data or available? Basically, under the 505(b)(2) application, it can be approved based on data from studies not conducted by the sponsor. That means you can have the agencies previously, you can refer to the agency's previously approved findings of safety and efficacy, so you can rely on another already approved product, or you can refer to clinical and preclinical studies from published data. So I think the last question we had is, do you need to do all the work? No, you don't need to do all the work. You can refer to what is already available out there. So what types of information are we talking about? So an applicant, of course, must identify all the sources of information they want to rely on for their for approval, NDA listed drugs, literature, OTC monographs. The applicant does not have to rely on a reference drug or a reference standard on one, but they can rely on more than one listed drug or more than one source of information. The applicant must also rely on a pharmaceutically equivalent drug if the pharmaceutical equivalent is approved prior to the submission of the 505(b)(2). So this is what the guidance says. The other topic that's addressed in the guidance is bridging/BE studies. This requires not only a successful bridging to the RLD or reference listed drug by means of either doing a comparative bioavailability study or a BE study. The pointer? Thank you. So here you can do you can show bridging by means of a relative bioavailability study and or a BE study, and but also some potential additional studies may be needed to fully support efficacy and safety of the new product. When BE criteria is not met, the number of adequate and well-controlled phase 3 efficacy studies will really depend on how close or how similar the concentration time curves are for the reformulated drug and the RLD. However, even if the BE criteria or BA or BE criteria is met, sometimes there may be need to still conduct some clinical efficacy studies and is required. And this are some of the situations where you may be required to have additional clinical efficacy study. Of course, when the BE is not met, when you're seeking a new indication, then you have to show safety and efficacy under that new indication to support efficacy of new route of administration or formulation for special population indication, for example, pediatric population. If you have or claiming superiority in your new oral fixed dose combination, or when you have non-inferiority posology bridging in new oral fixed dose combination, or in specific drug substance requirement, for example, in the opioid products. So what are the benefits of a 505(b)(2)? Benefits of a 505(b)(2) are in particular valuable for pharmaceutical and generic companies who are looking to alleviate competitive forces in the environment while wanting to benefit from a development process that eliminates most non-clinical studies as well as extensive safety and efficacy. So we're using the word eliminate, but we're using the word most. You probably will still have to do studies, but not as extensive as in the NDA. So the main 3 advantages that are listed are it's a relatively low risk because of previously approved drugs, so there's already something on the market. They're lower cost, accelerated development due to fewer studies, and I know this is good for the companies to save money. And the other thing that's of great benefit is you may qualify for 3, 5, or 7 years of market exclusivity, depending on your 505(b)(2) application. Whereas if you go to the generic route, it's a much, much shorter exclusivity period. So examples of 505(b)(2) applications, and this is really a little summary of what was presented by the previous speaker, because he went into a lot of detail about the different types of applications and what they entail. So this is just a summary. This I pulled from the guidance, so you can also go to the guidance and read the details as it pertains to all of these types of applications. However, I wanted to talk about bioequivalency as it relates to what we have been discussing the last the last day or so. And also, I wanted to say with Dr. Bloom's presentation, as he was giving the presentation, at the end I was kind of sad because nothing really happened to that product after all the work. And my expectation was his last line was that we submitted an application and this is their approval and they're making millions of dollars. And I was, oh no, you know, it's really, the suspense just killed me, literally. So I was very sad. But let's go back to the guidance and see what it says about bioequivalency. And it kind of clarifies based on what you were talking about. So bioequivalency, these are applications for proposed drug products where the rate and extent, and we saw that in your example, of absorption exceed or are otherwise different from the 505J, which is the ANDA standards for bioequivalence compared to the listed drug. And when you have the situation, if the absorption exceeds or otherwise is different, so we're not using the word failed, which was, you know, failed bioequivalence studies, depressing word, but it's just different, and we saw this difference. You can submit, this can be submitted pursuant to the 505B2 of the Act. Such a proposed product may require additional clinical studies to document safety and efficacy at the different rate and extent of delivery. Generally, the differences in the rate and extent of absorption should be reflected in the labeling. So whatever we're seeing, the difference we're seeing here, this should also be addressed in the in the labeling. Sorry. The proposed products does not need to be shown to be clinically better than the previously approved product. However, a 505B2 application should not be used as a route of approval for poorly bioavailable generic drug products unable to meet the 505J standards. So this is a pathway to use, but if you look at your product and you see it's just not a good product, don't submit it because it's going to be rejected. But for example, in the situation that was discussed earlier in the beginning of the day, I felt like there was sufficient evidence, and I think company protocol stumped the development of this product. So if the proposed product is a duplicate of an already approved product, it should be submitted. It should not be submitted as a 505B2 and should be submitted as a 505J. And if it fails or is has poorly bioavailable, then it needs to be reformulated. So, for example, in a 505B2 application, could be appropriate for controlled release product that is bioequivalent to a reference listed drug where the proposed product is at least as bioavailable as the approved pharmaceutically equivalent product, unless it has some other advantages such as smaller peak trough ratio. The patent of release of the proposed product, although different, like in the situation earlier, is at least as favorable as the approved pharmaceutically equivalent product. So here it's just stating that you have to show some advantage in relation to the approved product. I feel like I'm going really fast. So I'm almost to the end of my presentation. So what I've just discussed is some snippets from the guidance, from the 505B2 guidance on important topics and points that need to be understood when you're thinking about going through this route. The guidance is way more extensive than the few slides I've given. So my advice is to go back to the guidance and then see where you are in your situation. Now I wanted to give some examples of noteworthy 505(b)(2) submissions that have been submitted and approved. And when you get this, when you get the presentation, if you click on this, there will be 7 examples. In my presentation, I'm only offering 3 examples. So let's look at example number 1. This is Omitrope, and this is a noteworthy 505(b)(2) submission because it's a human growth hormone and is one of the early follow-on biologics that was approved by the 505(b)(2) route pathway before Congress and FDA put a biosimilars program. So even before the biosimilars program, the company thought to use this 505(b)(2) route, and they relied in part on previous findings of safety and efficacy of genotropin. Okay, so there was a product on the market. They relied on the safety and efficacy of this product, and they were able to be approved in 2006. And what are the changes that were submitted in the 505(b)(2) application? Was composition of the active ingredient changed, and what data or basic study requirements they relied on. They conducted BE study where they evaluated they did chemically comparative studies between the 2 actives and showed similarity in rate and extent of absorption compared to the RLD. So another example of a noteworthy 505(b)(2) application is Narcan. And Narcan was submitted by Adap Pharma in 2015, and they proposed a change to both the dosage form and the route of administration. Basically what they did was they converted the drug product from an injectable into a nasal product, and the applicant relied upon a relative bioavailability study in healthy subjects. However, because the injectable Narcan had been discontinued, ADAP used an injection of the comparator to bridge the FDA's previous findings on Narcan. So you can see here, there was an injectable product, it was discontinued. This company had the idea to make it into an intranasal product. So as you can see, they changed the dosage form and the route of administration. The basic study requirement was a relative bioavailability study and had to have a phase 4 commitment. And I think here you can also see that they did get a priority review for this product because the reference listed was discontinued. The third and last example is for Yasprala. This one is a special one because it has it seems very basic, but then it's a really good idea. So I'm going to read from what the publication says. We conclude with a 505(b)(2) that is noteworthy because what it combines and what it yields. A prescription, a fixed dose combination of 2 drugs that are one, very old, that's available over the counter as a monotherapy, and available, and also number 3, available in a cardiac event prevention strengths, and one of which may deliver pain relief even though it's not indicated for pain, and it potentially makes the drug safer. So Yasprala is a combination of 2 common generic drugs, aspirin, which is something we find every day in our pharmacy or even in our medicine cabinets, and an antiplatelet agent, omeprazole, which is a proton pump inhibitor. It is indicated for patients who require aspirin for secondary prevention of cardiovascular events, and who are at risk of developing aspirin-associated gastric ulcers. So this was a combination product that used something that was already on the market, old product, to provide more benefits to the patients. So it's a new combination. PK/PD and 2 clinical studies were conducted in 520 patients due to the combination, and that's what they use for the basis for their submission and approval. And that brings me to the end of my presentation. I hope it was informative, helpful, and also a refresher from what we had last year.
Thank you. Very enlightening morning this day, especially for a scientifically poor CEO like me, and technically very primitive. And the only thing that I will do later on is to ask my technical team to bring all those failed bioequivalence studies and make VAM out of them. So that's bring me to one question for Dr. Luis. Anybody have a question? We will, but because of the short of the time, I think we will be kind of a 10 minutes, I would say, discussion, open one. But let's see if there's one guy. I have one. So for the last example, what's the exclusivity that they gave them for this product? Do you know that, Luis? For the aspirin, omeprazole?
No, I'm not sure about the exclusivity, but I can find out and let you know.
Thank you so much. So let's invite our last speaker in this session, Dr. Rima Al-Atoum. I think everybody from the industry in Jordan knows Dr. Rima. She holds a master's degree from the University of Jordan, and she is the chair of the Bioequivalence Technical Committee here in Jordan. She is going to tell us about the prospects of VAM in the Jordanian FDA. So we are looking forward to.
Good morning. Today I will talk about Jordan FDA Value-Added Medicine Guideline. Okay. It's clear? Yeah. Here's the history of value-added medicine in Jordan. Now in the past there were many applications in value-added medicine in Jordan, but they were evaluated as case by case because we don't have a guidance. After that, Value-Added Medicine Draft Guideline was done in January 2018 and was published for comment in March 2018 and was finalized in July 2018. Here's our regulation. تعليمات تسجيل الأدوية المطورة ذات القيمة المضافة لسنة 2018 Value Added Medicine. Now, what's our definition of value added medicine? It's a medicine that has been modified or developed from the reference medicine, where the amendment would consider the medicine with a technology that increase effectiveness or safety. So you increase effectiveness or safety of the reference product or existing products. What's the objective here? To encourage innovation and to meet the unmet needs for the patients regarding safety and efficacy. In the United States FDA, there is a similar applications as Lois said, it's 505(b)(2). What's 505(b)(2)? It's an application that contains full report of investigation of safety and effectiveness where at least some of the information taken from other product from the reference. Also, you can find in EMA guidance, there is similar guidance with hybrid application. What's the hybrid application? Where a strict definition of generic medicine is not met. So it's not a copy, it's not a generic, exact generic. Where bioavailability studies cannot be used to demonstrate bioequivalence, for example, in case of locally acting products or inhalers or others. In our guideline, locally acting products, if they are pharmaceutical equivalent, it will be generic, not hybrid application. Where there are change in active substance, therapeutic indications, strength, pharmaceutical form, or route of administration of generic product compared to the reference product. So there is some modifications to increase safety or efficacy or compliance, etc. Now, according to our guidance, here you can find some example of value-added medicine. Route of administration, for example, going from oral to parenteral or transdermal or vice versa, or for example, to bypass, first-pass effect, it depends. Strength, for example, in case of supra-available and you want to decrease the strength and to be still bioequivalence, it could happen. In case of addition of a new indication, and in this case we need a clinical study to prove your claim. In case of fixed dose combination, it's included in our regulation, fixed dose combination. In EMA, I think not. It's not within the hybrid application, it has separate guidance. In fixed dose combination, yes, it's candidate for value-added medicine, but it's not including new chemical entities. By the way, value-added medicine in Jordan is not include new chemical entities and not include biologicals or biosimilars. They have different guidance. Addition of natural products, vitamins to the drug, for example, it's applicable. Change dosage form, for example, from tablet, capsule, or dispersible with justification. Active ingredients change, for example, using metabolite or prodrug, again, with justification. Maybe you go to different salts, isomer, complex, chelates, pharmaceutical alternatives. Formulation, for example, developing of modified release from immediate release, it's practical. Now in Jordan FDA, we highly recommend that the applicant ask for a pre-submission advice. Please ask for pre-submission advice before developing value-added medicine. And the pre-submission advice should include the rationale and the claim. What's your rationale and the claim? All data and plans, proposal, etc. It should be very clear. And all the supportive documents and published literatures, for example, that you rely on. So, it will start from pre-submission advice. You will ask the authority pre-submission advice. You will give out the idea and all the findings. And then there's electronic submission of the dossier from Module 1 to Module 5 according to our criteria. After that, if it's considered as a value-added medicine, the dossier will be studied by the originator committee in Jordan FDA. And in this case, if we consider it as value-added medicine, we will give you priority. This is a decision of the committee itself. And then, at the final decision of the registration and the pricing will be taken according to our registration and pricing criteria. And you may have privilege in pricing. So, regarding CTD modules or STD, in Module 1, it's according to adopted CTD checklist. Module 2, it's according to adopted checklist. Module 3, you have to concentrate more on pharmaceutical developments because it's key point. Module 4, according to checklist. And in Module 5, again, it's according to checklist, but it depends on your case. It's case by case, really. You can submit, or it may be essential to submit some pharmacokinetic, dynamic, clinical study, bridge, clinical. It depends on your case. It's not a must to submit all of these, but it depends. In general, the number and type of studies depend on several factors. intended use and patient populations. What's your indication? What's your claim? Again, what's the targeted group? Dosage and dosage form, dosage regimen. You have to prove what you included in your SMPC. Physiochemical properties and overall quality of the products. Here you can find the decision tree for submission of the dossier in Jordan, where to go. First of all, you have to ask a question. Is this medicine the reference product worldwide? Is your medicine as a reference? What's this mean? That you have a full dossier from Module 1 to Module 5, you have preclinical and clinical data. In this case, you will be the originator for Jordan, and it will be studied and evaluated by the originator committee. On the other way, if you are not the reference worldwide, then there is another question here. Is the reference product registered in Jordan? Now, if no, but you have a reference product, there is a reference product, maybe an EMA, maybe an FDA, or worldwide, but it's not still registered in Jordan, then really you are a generic. But we called it in Jordan FDA new drug. New mean you are generic, but new for the market, because we don't have information in the market from the originator itself, but still it's present worldwide. What we need in this case, we need published literature from the originator only, and to conduct bioequivalence or even sometime weathering, it depends on the PE criteria. And it will be studied again by originator committee. Now, if the answer is yes, there is a reference product in Jordan, then there's another question. Does the reference product in Jordan or even worldwide, same concentration, same salt, same dosage form, route of administration, which means pharmaceutical equivalent? Now, if yes, then you are simple generic. You are a copy, simple generic, and we need BE according to BE criteria or labeling system, and it will be evaluated by generic committee. But if no, you are not copy, there's some amendment from the reference, then you are a candidate to be as value-added medicines. It will be a committee decision. In this way, it could be you have to submit published literature, clinical study bridging, bioequivalence, pharmacodynamic, it depends on your case, and again, it will be evaluated by the originator or the concerned committee. Here are some cases of hybrid VAM, fixed dose combination, scientific advice. They are submitted after the guidance during the last year only. Now, before that, yes, we have a lot of cases, but it was case by case evaluated, as I mentioned before. Now, forgive me if I don't mention the name of API in some cases. It's still confidential. It's for local companies under discussion. This is a simple case. The MAH has submitted application to register sildenafil citrates 100 milligram, 50 milligram, 25 milligram chewable tablets. Now, as we know, Viagra is the reference product. It's film-coated tablet, not chewable tablets. What's the rationale? More convenient for some patients. Okay. For the generic application, the MAH has submitted just simple bioequivalence study on fasting states at higher dose, 100 milligram. Now, what was the decision of the committee? As you see, it's some modifications, some simple modifications. Instead of film-coated tablets, we got the chewable tablet. Yes, it's more convenient for some patients, that's good. Or, for example, oral dispersible tablets. It can be within the definition of pharmaceutical alternative, according to EMA, or according to FDA, petitioned ANDA. So, based on the result, 90% confidence interval were accepted for AUCG, AUC Infinity, and Cmax. It's within the acceptable criteria or limit of confidence interval between 80 and 125%. So it's acceptable. And also wavering can be done. Extrapolation to different strength as they met the condition. There's a linear pharmacokinetic, there's dose proportional, same pharmaceutical conditions, etc. So it was applicable to make wavering, but the product authorized in Jordan and marketed in Jordan as a generic. It's a simple generic pharmaceutical alternative, patent AND. It's not a value-added medicine, and it's not considered as VAM, but still, you have advantage of authorization. We authorize the product. You can take the benefit from the market, which is good. UNMH has asked for scientific advice if they can develop oral jelly medicine product for systemic use, taking in consideration that the pharmaceutical alternative. This was the claim in the beginning only. It's pharmaceutical alternative. Instead of giving the reference in film-coated tablet, we will take it on oral jelly. That's more compatible or is more convenient for some patients as a reference in film-coated tablets, not in jelly. So, what were they asked in the beginning? They asked us, there's really another generic or similar products worldwide. It was, I think, in India, in jelly preparation, or in China, I don't remember. And in the beginning, they asked us to make equivalence with other generic formulations worldwide, not the reference. What was the committee decision? The BE study should be conducted between the test jelly preparation versus the worldwide reference. So it's not allowed to make a comparison with other jelly formulations. If the reference is not within this formulation, you have to go and compare yourself with the reference in tablet formulation. And besides that, the company should prove the safety of the volunteer before conducting the study, just to ensure that there is no potential risk of buccal absorption that may increase the risk and adverse effect for the product. What happened next? The applicant conduct pilot BE study with reference as a committee recommendation, that's right. Now the results of AUCT and Cmax exceed the acceptable criteria. It was more than 80 to 125, so it's not equivalent. And the Tmax was lower than the reference. Now there's a new claim. What's the claim? We have higher and faster efficacy, and it's desirable for some patients, according to their claim. Good. And they bring some of published literature, but it wasn't enough, it wasn't enough. Okay. As the applicant claim now that their formula get higher efficacy and faster effect, then we need pivotal BE study and we need clinical study, which should be submitted, because you claim now that you are more effective and you are faster. Again, we want the SMPC, your clinical study should be in line with your claim and in line with your group, in line with your dose and dosing regimen. Meeting with committee is always applicable, and we can discuss the proposal and the requirement, and we already scheduled meeting. Another case, MEH asked for scientific advice if they can develop product with double the concentration, 10/160 milligram worldwide reference product, nebivolol valsartan, which is marketed on 5/80 milligram, claiming that it's proved to be safe and effective in some literature for this design of its strength. Now, what MEH asked, he has only proposed that it's sufficient to conduct bioequivalence study comparing one tablet of test versus two tablet of reference. We always review all the findings and we make our own search during the pre-submission advice. As reference products in the lower strength, 5/50 milligrams, still not registered in Jordan, but fortunately it's listed in United States FDA, so it's RLD. Referring to the worldwide reference products label, it's stated clearly in section of dosage and administration. The recommended dose of RLD is 1 tablet with 5/80 milligram. Dose taken orally once daily maximum effect are attained within 2 to 4 weeks. Now, it was clear. Increasing the dose does not result in any meaningful further effect. See clinical study of the RLD. So, is it applicable? Based on the clinical study stated in the reference label approved by United States FDA, there is no clinical benefit of increase the strength. It's already done by the reference. So the application considered not applicable. I know there's nothing impossible with science. That's right. But I think it's a very hard situation to defeat a theory already proved by the reference. Other case, applicant asked for scientific advice for clinical requirements and for waiving request for the following products, simple products, phenylephrine, fixed dose combination paracetamol with phenylephrine, and fixed dose combination phenylephrine with loratadine. And the applicant claimed that they are OTC products, that's right, and not require any bioequivalence study in country of origin. It's an important product. What was the committee decision? Now regarding the first two, phenylephrine and the fixed dose combination paracetamol and phenylephrine, yes. They are OTC products and they are well established in Jordan markets with no bioequivalence problem in conventional dosage form. You can apply wavering for these products. They are like grandfather products. But regarding the third one, phenylephrine and loratadine, as we don't have such combination in Jordan, it's not marketed. And if there is any international reference, I doubt, in EMA or FDA, then it's simple generic and you can conduct BE study. But if not, then you are fixed dose combination. You can refer to a helpful guidance in EMA guidance of fixed dose combination. You have to state your claim, your evidence base. What's your scenario? Is it add-on? Is it substitution? Is it initial combination? I don't know. And according to your claim, you should submit clinical, pharmacokinetic, dynamic. It depends on your claim. Another case, or scientific advice, MAH asked for submission, application of fixed dose combination product containing paracetamol, thiocolchicoside, based on wavering from BE claim that both API are BCS class 3. It's an important product too. Now this combination not authorized in the United States. We don't find RLD. If it's not authorized, I think, in EMA as a reference product, we don't find that. Now based on the fact that we don't have reference product in Jordan or in other, but especially in Jordan, we don't have reference product. So in all cases, BCS wavering is not acceptable as BCS class 3. There's no evidence, and we don't have any information about the formula of the reference, how to waiver it based on BCS class 3. They may actually submit again, so you are, according to our guidance, you are fixed dose combination, and you can refer to international guidance of fixed dose combination to show your claim, evidence base, what's your scenario, and to submit the desired data. Another case, it could be applicable. Applicant asked for requirement of submission combination of API with multivitamins. There is no international reference for such a combination, and they claim that it's already authorized in country of origin based on some articles. They don't ask for anything. No further information submitted from the applicant. Again, the same decision. What about your SMPC? What's your claim? What's your indications? What's your age group? I don't know. You are a new fixed dose combination regarding to Jordan. We don't have reference worldwide or even in Jordan. So you can refer to the requirements of new fixed dose combination. Another case, applicant asked for developing fixed dose combination based on substitution therapeutic scenario. Which is good. I think we have 3 similar cases like this. Now, the reference MPC stated clearly that drug A can be taken with drug B at a designated dose. Thank God. Applicant offered a proposal of BE design to compare co-administration of both reference monotherapies versus the developed fixed dose combination. It's rational. And there's many references from peer-reviewed articles submitted as a proof of safety and efficacy for the designated fixed dose combination. Perfect. Now, based on all the submitted references, the application considered applicable, which is good, it's applicable. Now, when the authority says it's applicable, this is advantage for you. That means we can authorize this product. But is it with a value? It's a question mark. It's applicable, good. Maybe it's not value-added medicine. We asked for a drug-drug interaction study or literature for that. Further discussion need to determine the real value beyond development of the proposed product regarding priority and pricing. Also, always our door is open. Meeting with committee can be asked to discuss the details. Now, in general, substitution practice is not favorable to be a candidate for value-added medicine, except in some cases it's very essential, for example, for geriatric, pediatric, or cost-effective, it could be. So if you have an idea, we highly recommend again to ask for advice. Now, so advice or pre-submission advice is not dedicated only for value-added medicine, it's preferable in value-added medicine, but you can ask for advice in any section in the dossier, especially, for example, for reference products. Don't start to conduct even simple BE if you don't sure from the reference products. I think there will be a session for the appropriate reference products. If you don't find reference products in the market, please come and ask the authority which the appropriate reference product to compare with. Again, referring to the VAM, if you have VAM idea, then you have to state your rationale, your claim, your proposed MPC, your indication, your dosage. We need this information. There is an international guidance support your claim or support your scenario. Do you have peer-reviewed article and literature review? Did you conduct any pilot studies? You have to set all these references together, and then you have to make a proposal. How to make a proposal? You can go and ask help from specialists, you can ask CROs, you can help, yes? CROs. You have experience in clinical studies. You can ask academia, you can ask universities, whatever you want. But you have to give a proposal. After a proposal, then you will ask for pre-submission advice. But never go directly from, I have an idea, please give me advice. It's not applicable. We will not make a protocol for you, and we will not make a proposal for you. We will discuss your proposal. As a conclusion, value-added medicine innovation is concerned to achieve advantage regarding extra value, regarding safety and efficacy to meet the unmet need for the patients. Not all hard applications can be considered as a value-added medicine. Depend. Scientific advice is very important. It's very, very important. It will help you, and it will be cost effectiveness, and it will save your money and time. The scientific advice should be clear, specific, and including all the supportive information. Your rationale and claim is a key point. Your clinical studies or your studies, it depends on your claim. For fixed dose combination, you can refer to the EMA guidance, for example, or other international guidance. You have to set your claim, evidence base, your scenario clearly, and you have to submit either pharmacokinetic, dynamic, or clinical. It depends on your claim. And thank you for your attention.
Thank you, Dr. Rima, for the insightful from the JFDA. I think we have 10 minutes. So if you'd like, we can make it, we can ask all our speakers to come to the stage, or make it very casual. So we can start by this question. Do we have another mic?
Thank you, Dr. Rima, for a nice presentation. Just simple question for the point, for the case you pointed out regarding the addition of vitamins with the API. I think if the dosing for vitamins are below the RDA, I think this is nothing to do with that, because these are usually considered as food additives. Are you mean that for vitamins with higher doses, or
I agree with you. Yes, it is lower than the RDA. It will be not a VAM maybe, but we will ask for some other information regarding if there is interaction maybe, or if there is something to read about. It will not just simple generic. We need other information.
Excuse me. Yes. Sultan al-Dayr. Here, Dr. You are looking for me. Sultan al-Dayr speaking. I have a question about the scientific advice. First, I would like to thank the JFDA for issuing this guidance, which will open many doors for the generic companies in the Arab region. And I know it is the first of its kind, and we hope that all regulators will follow you. So, regarding the scientific advice, is there a fees for the scientific advice? There is.
Up to date, fortunately for you, it's not up to date. But we have now a draft for scientific advice, and it will be published soon. And yes, we will specify a fees, but up to date, no. Just headache for authority.
So that's totally fine. But what about, is the scientific advice open for all companies, not only for the Jordanian companies?
Absolutely.
Okay. Let's assume that we have a VAM, and since the JFDA is the first to issue this kind of guidance, we are a Saudi company. Can we submit our products first to the JFDA?
No.
It has to be approved in the country.
Except if you make contract with local company to be a marketing authorization holder. This is our regulations. Okay, you can go with contract.
Thank you very much. I've been listening for the last 3 sessions. I would like to say, I'll tell you, because we are in Europe, maybe you haven't mentioned, I think Dr. Rabab said on the phone, there's something called value-added medicine. And I said, what do you mean by value-added medicine? We don't have that definition in Europe as regulatory affair, or as a regulator. We don't have that. What we have is actually, we call early access medicine scheme. If you look into the EMA website, they call early access medicine, EAM, which is exactly. And that actually tell you in details what you need to do if you have a value-added medicine. And it will have a fast-track access.
We get priority too. It's value-added medicine.
I don't know what EMA, I'm talking about
that, yeah.
People who would like to approach Europe, who would like to bring their product to Europe. There is early access medicine scheme. I was involved in the designing the quality side of it, and of course the medical doctors were actually involved in the clinical side. And we have set the procedure exactly what to do, and how long will it take, and the stages of the advice. We'll send you back to do the work as agreed with the committee, and then once it's been approved, hopefully we'll be there. And that will, as Dr. Rima said clearly, we're not considering about hybrid. The hybrid is 10-3, and is defined by the guideline what hybrid 3 is being mentioned in the guideline. Early access medicine is completely different thing. For example, like insulin by inhalation. There is a medicine now licensed by FDA, I think 5 years ago, for insulin by inhalation. I haven't seen it come to EU because of difficulties. I don't know, somebody from the FDA can tell us about it. So there is actually a very good opportunity for the innovators, regardless of Jordanians, Saudis, everywhere, can access to Europe within a very short period using that scheme, and it is very helpful, nicely designed, and very easy to do as long as actually you have the right product for the right indication. The other thing, there's a scheme for the orphan indication, and that will give you, of course, exclusivity for 10 years for both of them. The orphan, when it is actually an already licensed medicine or a non-active, but never been actually approved or assessed for orphan indication, and in this case, there is, of course, acceptable criteria of what to apply, what to define by orphan indication. For example, as I was with MHRA, we just approved caffeine for an orphan drug. Caffeine, the one that we create. So there is an access in medicine for the value-added medicine in Europe. Thank you very much.
Thank you, Dr. for the advanced information. Thank you.
Dr. Hakim.
Thank you. I don't know. My question is for Dr. Lewis. I've noticed that in the example that you've mentioned, the first two examples, although the first one was the growth hormone and the other one is, and only what was required is a bioequivalence study, and even have been got a priority and exclusivity, which is really in fast track submission. Can you please highlight something regarding this? Because we need to understand the maximum requirement that should be asked. What are the requirements? Especially we are talking about well-established product, well-known safety, well-known efficacy, and all the hybrid idea is based on safety and known. Yeah.
That would be the last question.
Hello. So in terms of what I presented was a summary of the main information that was used. Now what I did present, or what you did see from these examples, is how much information did the agency have for the product that was being relied on. And maybe that would speak to how much information was already available that the company didn't need to do so much. So with that information, I really don't know, because I wasn't part of the review process, but I maybe can extend a comment from Dr. Suleiman, because he was in the new drugs office, and he can maybe talk about how much information and what determines what you submit to the agency. It's really based on what is already available or what you can submit from at the agency or in literature.
So implementations, I'm actually going to answer your talk offline further. Typically, from the agency's perspective, usually with the 5.5b2, we really are filling the gap, in essence. So you're filling the gap. I think there are general guidelines that are there, but quite often with 5.5b2 applications, the divisions have a lot of leeway to basically ask anything that they may feel is missing. I've been involved in 5.5b2 products where companies have been asked for non-clinical toxicology studies, which is not common, as well as other atypical kinds of data. For opioids, actually, they would ask for risk management plans as well. A lot of things that are not typically asked for generic products, even more so. So it's really as I said, yeah. Again, I think to Dr. Los's point, it really depends. Again, there's a huge range, and it's by design. Really, if you read the 5.5b2 guidance, it just basically states that you rely on the findings of the preferences of the drug in terms of safety and efficacy, and you can rely on publicly available information, which really means you really have to fill whatever gap there is. And in some cases, with grandfathered in drugs or opiates or other drugs that have been approved 50 or 60 years ago, there's a huge gap.
So, again,
I find this is unfair that we don't make a question for Professor Heaney. Therefore, I have a question to him, please. Your presentation was very excellent. Once again, as usual, I was a student sometimes in your lecture in Frankfurt. I would like to ask you how you made that drug which is with cholesterol reducing agent that is receptors is saturated. I don't remember that at all. That means that distribution phase in your pharmacokinetic is not too long according to the Michael-Menten equation, or what is really working as reducing the dose to 40 milligram. Why you don't reduce this at the first to 50 milligram, and that you know how this the receptor is saturated, and means how to come to this again?
Well, in the interest of time, and it is my primary job now to make sure that you go for dinner, for lunch, sorry, in time, so that we have the chance for this session, which we postponed yesterday, this afternoon. And therefore, I would suggest that we discuss this during lunch break. I would like to make a few comments on the last session, starting with the presentation of Attila Hinschau and Dr. Eber's presentation. I enjoyed both, and of course Louise as well, but this has been already discussed. I really enjoyed your presentation, and I think this is a very, in my eyes, very appropriate approach Jordan FDA is suggesting, and I was very happy to see that. I have a slightly different position in one particular case, because I think if you develop the first fixed-dose combination of a combination which is used as free combinations in practice, is an advantage, because you don't need two different drugs to be taken at the same time. You take just one. It is an advantage. If you talk about an advantage, chewable tablet compared to swallowable, then this is also an advantage in my eyes. My comment to Europe is the hybrid application is the equivalent to the 545(b)(2) approach, if you follow it very carefully. And we started a year ago with exactly one example on enteric-coated PPIs, where more or less the innovator product has a significant food effect, and all the generic development products has a clearly lower food effect. And the food effect was a reduction in systemic exposure in the innovator product. And all the generics had higher bioavailability in fat state compared to the innovator, and were bioequivalent in fasted situation. This is an advantage. However, they are not generic. They are not, of course, bioequivalent in both scenarios. However, they are applicable and they are provable based on hybrid application. Therefore, I think to say hybrid application in Europe is not a way to added value medicines, I disagree on that. It is an value added medicine under these considerations, and therefore this is the approach in Europe. And for example, the situations for others, for example, for products with only very, very few patients, this is not the discussion we have today. If I summarize, or if I look at the examples which have been shown, I think there are certain examples which are pretty easy to be developed and improved. That is, for example, give really a super bioavailability. I don't talk about 10% more bioavailability, but if you increase the bioavailability, and I showed an example out of the field of anti-diabetic drugs, the new medicines, the new compounds which have been developed, and there you can increase the bioavailability by a factor of 10. And if you then reduce the dose to 1/10 and has the same efficacy, then this is clearly an improvement. Therefore, this is a field. And the fixed dose combination, I have already mentioned, the substitution indication, and the single enantiomers may also be something which you may consider, because you do not need the full clinical program in certain cases, but measure just the active enantiomer, and if this is developed instead of racemate, this may be an advantage as well. So there are cases which are less comprehensive in terms of the needed clinical data to support this application. And this seems an appropriate way, and if you follow the advice of Dr. Lema to go for the discussion with the committee, and I fully agree with her, I always advise companies, don't go there to ask questions, go there to defend your concept. You know, you have an idea, you have a vision, you know the product better than the authorities, or it should be that you know it better, and then take all your arguments together to try to convince them that this is an appropriate approach. This is the way to go, and I understood that this is also what the agency expects, and I'm fully on your side. Thank you very much. Now we have, let's say, 45 minutes for lunch, and please be back in time because we have to give time to those